This is about chromosomal basis of sex determination in humans. So this is father with the XY chromosomes and mother with the XX chromosomes. So these are the children produced, offsprings produced. So X, 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 and XY and XY. So this is how children are born, and so this is the determination of sex in humans using this chromosome type. And so this is sex determination in birds. This method of sex determination in birds is little different, because in birds both sexes, male heredity and female, possess two sex chromosomes, but unlike human beings, the female has the heteromorphic or heterogametic, that is having different sex chromosomes ZW, while the male, it bears the sex chromosomes ZZ are homogametic. So it is opposite of what is seen in human beings. So here females are heterogametic and males are homogametic. Thus females are heterogametic and produce two types of eggs, that is A plus Z and A plus W. Here A, it stands for autosomes. The male gamete is only one type, male gametes of only one type, that is A plus Z. This type of sex determination is called ZW, ZZ type, or WZ, ZZ type of sex determination. The letters Z and W are used to distinguish these types of sex chromosomes from XY chromosomes found in the XY type of sex determination in human beings. So we shall move on to this diagrammatic representation, how sex is determined in birds. So this is male AA plus ZZ, in female it is AA plus ZW. So these are the parents and these are the gametes produced, A plus Z, A plus Z. Okay, and here it is A plus Z, A plus W. See, these are the gametes from male and these are the gametes from female. So now the combination, that is offsprings, it shows A, Z, Z. Then A, Z, Z. And next one, it is A plus Z, W. Likewise, A, Z, W. So these are females and these are males. And this is about sex determination in honeybees. It is quite interesting to learn about how sex is determined in honeybees. Honeybees have a unique method of sex determination. In honeybees, fertilized eggs emerge as females, that is worker bees and also queen bee. The unfertilized eggs develop into males. Since fertilized eggs and also females are diploid and unfertilized eggs and males, that is, are haploid, sex determination in honeybees is referred to as haplodiploidy, sometimes also called arenotoky. The sex is determined by the number of sets of chromosomes an individual receives. For example, the male, which is called a drone, male is called a drone, it is produced from unfertilized haploid egg. Okay, so no fertilization at all, unfertilized egg, it develops into a drone or male bee, and thus male honeybees contain a single set of chromosomes. Because only the haploid egg, so single set of chromosome, it develops into male honeybee. The female honeybees, which are worker bees and queen bees, are produced from fertilized egg and therefore are diploid. They contain two sets of chromosomes. In this case, only females are produced by sexual reproduction. So females only are produced by sexual reproduction. Males are drones are produced by not sexual method, because unfertilized egg develops into males or drones. It is very interesting in honeybees that males have no father and cannot have sons also, but have a grandfather and can have grandsons also. This is interesting to note down. I am repeating. In honeybees, males have no father and cannot have sons, but have a grandfather and can have grandsons. So this problem, its a solution is also given. It's not so important. If you want, you please go through. So we shall move on to the next slide. It is about criss-cross inheritance or X-linked inheritance. The genes which are located on X chromosome, that is sex chromosomes, are called sex-linked genes. These genes show criss-cross inheritance. When a male has a defective sex-linked gene located on X chromosome, he transmits the defective X chromosome to his daughter during reproduction. So remember, a male has a defective sex-linked gene located on X chromosome. He transmits the defective X chromosome to his daughter during reproduction. The females who have this gene transmit it to her son and daughter both in equal probability. So the male passes on his recessive sex-linked trait to 50% of his grandsons through his daughter. The sex-linked trait being recessive is not expressed in female, but it is expressed in males. Therefore, males suffer from the genetic defect due to the presence of faulty gene on the single X chromosome, while females are only carriers of these defective genes as they have the other X, which mask the effect of faulty gene. Hope you understood this. Females, they have XX chromosomes. So any one X carrying the defective gene will not affect them, but they are said to be carrier females. But in males, because one is X, other one is Y, so this X chromosome, if it carries defective gene, then the father or the male will definitely pass on this defective gene to the offsprings. So there is a chart also we will discuss it now. So the trait shows up in female only when both X chromosomes from mother and father have faulty gene. So females are the impact of this sex-linked chromosome on females is very less because females have XX chromosomes. Only when both the X chromosomes, if they have faulty gene obtained or received from mother and father only, then in that case only females will be affected. So this is a chart, diagrammatic representation. So this is a carrier female and hemophilic. Okay, hemophilia is a genetic disorder. You know it. So this is a carrier female. So this XX chromosomes, one X carrying normal gene, other X chromosome carrying defective gene, small h, and this is normal is capital H. And this is a normal male. So X, capital H, normal. Y, no defective gene or that sex-linked, that defective gene is present only on the X chromosome. So Y has no defective gene. So these are the gametes. X, capital H, X, small h. Okay, these two. Here, X, capital H, and Y. So this is the Punnett square at Punnett table. So this shows this is X capital H, X capital H. So this is a normal female, and this is X capital H, X small h. So this is carrier female, and here it is X capital H, Y it is normal male, X small h, and Y it is a hemophilic male. So this type of inheritance of recessive sex-linked character from father to daughter and then from the daughter to her sons is known as criss-cross inheritance or sex-linked or X-linked inheritance. So this is about criss-cross inheritance. So we shall move on to the next one. This is criss-cross inheritance in humans. This is about color blindness, how it is inherited. Okay, here also the defective gene is located on X chromosome. Thus a single defective gene causes disease in male, while two defective genes, homozygous condition only, can cause disease in female. So the probability of this disorder in females is reduced. Okay, so only in homozygous condition, if both the X chromosomes carrying defective gene, that will affect the females. Female is heterozygous condition. Females in heterozygous condition are apparently normal, but actually they are the carriers of the disease. The disease is expressed only in males because of we have the partners of the genes on Y chromosome to mask the effect of the faulty gene. Okay, the Y chromosome, X chromosome is only one, and Y chromosome, so it cannot mask the effect of the defective gene. This is the diagrammatic representation to show the inheritance of a defective gene responsible for color blindness. So this is the female, not affected, but at the same time carrier female, because one of the X chromosomes carries this defective gene small n. So this capital N refers to normal gene, small n refers to defective gene, and this is the male unaffected because X carries normal gene capital N and the other one is Y. Okay, so these are the parents. So these are the gametes, X capital N, X small n. Okay, these are the gametes produced from carrier female, and these are the gametes produced from normal male, X capital N and Y. So the combination of these gametes produces this individuals in this ratio. That is X normal N, that is capital N, X capital N. So this is normal, not affected, unaffected, and this is the carrier and not affected, because one of the X chromosomes carrying normal gene capital N, the other X chromosome carries defective gene, so small n. And this is the unaffected male, because X having normal gene capital N, so this is normal male. The other one, this X chromosome carrying defective gene represented by small n, so this is colorblind male. Okay, so this is about color blindness. So in any combination you can get this, that is, you can, instead of this carrier female and normal male, you can change this also, you can change it into normal female and affected male, like that. Okay, anything you can. So accordingly you will get the offsprings in those that is as per that ratio. And so this is the last slide. This is again repeated about hemophilia, so you can go through.